Hi Bio30, welcome to chapter 17, looking at genetics and how traits are inherited from parents to offspring. We're starting off with a few interesting genetic conditions that you can check out in my links. Just make sure that you're opening the PowerPoint to access the links, as they won't open on the PDF version. The PDFs are included because those will not have any diagrams covering text, so you can look at notes for PDFs, but open links from PowerPoints. Yeah, take a look at some of these interesting conditions as we start off our genetics chapter. And genetics is studying heredity and how conditions and genes and traits are inherited from parents to offspring. And genes are located on the chromosomes, and they will be sections of DNA, all basically giving information about one trait about you. And this is just looking at some different dog breeds and how breeding is done to select for certain genes and genetic traits, and that's selective breeding. We start off genetics by looking at Gregor Mendel, because he is considered the father of genetics and studied many genetic crosses, particularly with pea plants. And sadly, his work was ignored until after he had passed away, but we still go back to all of that as the start of the understanding of genetics. And he used peas because they're small, grow quickly. He could cross-fertilize them, self-fertilize them, and they had a lot of traits that were where there were only the two versions of that trait, like height was either tall or short, or flower color was only either pink or white. So it was easy to do crosses when there were only two versions of many traits about of those particular species of pea plants that he used. So he didn't know about the word gene, but he called them factors. He came up with the principle of inheritance, saying that with every trait, you did inherit a gene from each parent. Now, when we look at chromosomes, I just drew this on the page here. Here's chromosome number 16, perhaps from dad, chromosome number 16 from mom. You do have two copies of each chromosome in every nucleus of every cell, and on that chromosome will be what particular a gene. For example, for tall, on dad's chromosome you might have gotten the tall gene, and on mom's you might have gotten the short gene. So it's for the same trait, but it may not be the exact same version of that trait that is contained in your pair of chromosomes for number 16. In the case of blue eyes here, this is just showing you could have gotten both recessive traits for blue eyes on both, and then you for sure would have blue eyes. If you got a tall and a short, they would be always exhibiting whichever is the dominant. So you would exhibit the dominant when there's one of each. Just a little warm up to how we're looking at what traits are exhibited. Now, in terms of pea plants, here's some of the traits that Mendel looked at. And remember that whichever is the dominant one, which they have put on the left side here, the dominant, those will be the ones that we use the letter of when we are using symbols for those traits. We'll use the symbol of the dominant one. The other one will be the recessive one. And so for pea plants, we would use T for tall and a little t for short. It has to be a lowercase of the same letter that you used for the dominant. For round, we'd use big R. Wrinkled, we'd use little r. Yellow, green, little y. We might use green for the color of the pod, and then the yellow pod would be the lowercase. So whichever dominant trait we're looking at, this is looking at the two possible pod colors. This is looking at the two possible seed colors, two possible seed shapes. Whichever trait that is, there will be at least two possible versions of that trait. And when we do crosses, we just look at two different plants that are being crossed and see what combination of traits their offspring will have. Now we'll use the letter P to indicate the parents that you're crossing, and the offspring will be their first set of kids. That's their first filial generation. So if we do a cross, for example, of, let's say, a round plant that's carrying a wrinkled gene and a plant that's entirely carrying wrinkled genes, this would be our parents right here. So those are the parents that you're crossing, and the offspring that you're going to look at from those two parents would be your first set of kids, your first filial generation. Okay, if you were to take two of those offspring and cross them again, that would be your second filial generation. You will be using the terms F1 and F2 as we do crosses. Now, a monohybrid cross is when you are only using one trait at one time, looking at one trait at one time and crossing it, whereas a dihybrid cross would be where you would, that's our next topic, a dihybrid cross would be where we look at two traits. So we might look at what this in one individual has for the round wrinkled and what they have for the tall short. And you'd cross that with the other one that had two wrinkleds and maybe a tall and a short. So if you're looking at two traits at one time, that would be a dihybrid cross. The word hybrid means that that individual does contain one of each. So if you have an individual whose genetic makeup is that one parent gave them the dominant and one parent gave them the recessive, a version of that trait, that would be a hybrid individual for that trait. With dominant and recessive traits, we know that the dominant trait will always be the one that's expressed. If there is a combination of traits, one dominant, one recessive, you will always have the expression of the dominant, and you would only have the recessive shown or exhibited if there are both parents have given you a recessive, then you will exhibit the recessive trait. The principle of dominance, we should know this, is that if you have two alleles that differ, the dominant will mask the recessive. So the two versions of a trait are called the two alleles or two versions. So this is round and wrinkled, for example. And dominance, principle of dominance says if you do have one of each, you will always exhibit the dominant one. So this plant would have round seeds, not wrinkled seeds. You would never know by looking at the seeds whether their parent gave them it was a plant that had round, because their seeds would be exactly the same round as if they were this combination of round wrinkled, because their seeds look exactly the same, because both of these are exhibiting just the dominant. And so by just looking at a seed, you could not tell its genetic makeup as to whether it's hiding this or masking that recessive and it is got one of each and it's heterozygous, or if it's homozygous dominant. So just by looking at it, you could not tell. You'd have to do some crosses with that plant to be able to determine what its genetic makeup is. So the law of segregation means separation, and so the idea here is that when your gametes are formed, your two chromosomes for each pair are going to separate, one into one gamete and one into the other. And so a sperm and egg would only have one copy of that gene. And so if the sperm has just, for example, the round allele, the egg, depending which one it has, because it only has one of each, it's a haploid cell, that egg may carry the wrinkled. And so when this new zygote is formed, the genetic makeup for that particular trait would be a heterozygous, and this idea of segregation just means that whatever the parent had, those genes will separate when gametes are formed. So there ends up to being only one allele in each gamete for each trait. The monohybrid crosses that we'll work on first are just looking at one trait at a time, as I showed you earlier. So looking at one gene, for example, the shape of that seed, whether it's round or wrinkled, you're looking at one gene that would be found in one spot on a chromosome. And remember, you have 2 of every chromosome, so you might have, let's say, recessive round in both spots. In both mom's and dad's chromosome number 7 gave you the same allele. That's possible. That would be the gene for maybe round or wrinkle, we'll say for the shape of the seed. And the alleles possible in this case is that there could be round or possibly wrinkled versions of that gene. The genotype would be when when you look at both of those chromosomes at one time and you see what combination they have, that would be the gene combination or genotype. Whereas the phenotype is the physical nature of that organism because of that gene combination. Physically, they will look this way. So with this gene combination, they will look round because they have one round allele, and round is dominant. So something that has a plant that has either heterozygous or homozygous dominant combination will be round. Homozygous means that there is 2 of the same, either 2 round genes or 2 wrinkled. Whereas heterozygous is the same as hybrid. It means there is 1 of each. Okay. Those are some terms that will be important to review and understand as we go on in crosses. So this is an important page. Make sure you know these terms. A Punnett square is these squares that are used to check out what possible combinations offspring will have. So, for example, in this question, one parent had a combination of heterozygous, 1 round, 1 wrinkled genes. So half their, for example, if this was the male, half the sperm would be carrying the round gene and half the sperm would have the wrinkled. If it was looking at the other side, the female, and if they had the same genotype, or they were also heterozygous, that meant half the eggs would carry a round gene and half the eggs a wrinkled gene. So in doing a Punnett square, we're looking at what combinations the offspring could have. And you just will combine them and say the offspring could have 2 rounds. The offspring, this one and this one, could be heterozygous but still round. We go over here. The offspring here could be heterozygous, still look round. And if both little recessives got together, a quarter of them could be wrinkled. So this just tells us that 3/4 of the time, 75% chance of the offspring of these 2 individuals will have round seeds. Whether they have 2 round genes or 1 round, 1 wrinkle, they will have round seeds in their offspring, 75% chance. And a 25% chance of having wrinkled offspring. So because it's 75 to 25, we say that it's a 3-to-1 ratio physically, phenotypic. Physically, their offspring will have 3-to-1 ratio of round to wrinkled. And that's how we use the Punnett square to see what the offspring are. Now, if we are looking at the same exact cross, and if someone has asked you to tell what the genotype of the babies are, that is different. If it was phenotype over here, you're just looking at physically what the offspring were. 3/4 would be round and 1/4 would be wrinkled. But genotype means what gene combinations there are. And you can see that there's the gene combination of both of the genes being dominant. There's the gene combination of one dominant, one recessive, and the gene combination of both being recessive. So if you do have a question that asks what is the genotype or gene combination of the offspring, you have to say that the gene combination is a 1-2-1. You've got one that's both the same homozygous dominant, you've got two that are heterozygous, and you've got one that's homozygous recessive. So genotype means the gene combination of the offspring. Phenotype means the physical look of those offspring. And the physical look of these offspring, it's the same cross, but the physical would be that there's 3 rounds to 1 wrinkled, whereas genotype, the gene combination, is that there's 1 of these, 2 of these, and 1 of these. It's a 1-to-1 ratio. And sometimes they may state it as 2-1-1, usually 1-to-1. The important thing about answering the questions is to tell, once you write a ratio like this, underneath write down what you are saying that that ratio stands for. And same with over here, physically they look 3-to-1, so you write down underneath that 3 would be round and 1 would be wrinkled. So you write down underneath what your ratio stands for. This is looking at a plant that has homozygous recessive two little r's. So that individual plant, their parent gave them both recessive genes, so it was a wrinkled plant, and both if it was the male plant, let's say, all gametes, all sperm were carrying wrinkled gene for that trait, because that's what their gene combination is. If this is, for example, the female plant, and it had round seeds, but half of them were dominant, half are recessive. If we do the cross for these, you see that in doing it, we get half the babies being round and half the babies being wrinkled. So in this kind of a cross, physically, we've got a 1-to-1 ratio of half are round, half are wrinkled. So instead of saying 2-to-2, we reduced to the lowest number, and instead of 2-to-2 is just not acceptable because you can reduce that to a 1-to-1 ratio. So the answer for physically how the babies would look in this cross is that they would have a 1-to-1 ratio, and you tell what that stands for. Half are round, half are wrinkled. Gene types are also in this case the same, 1-to-1 ratio of half of them being big R, little r, and half of them being little r, recessive, wrinkled ones. So you can see that there are just an easy way to tell physically what the offspring look like from a cross, and you can easily tell genotypically what the gene combinations are of a cross. And as we practice them, you will see that these are really easy to do. Probability is just looking at sometimes multiplying. If they ask you what happens the first time, if offspring were born, you have a certain amount, like 3/4 of the time it would be this, for example. And if they say in a row, then this is the key word here. If they say what happens if you get what's the chances you get, for example, blue-eyed child 2 times in a row, you would just multiply the answer that you got. So if you have this example here, it says if you toss a coin and you get heads half of the time, when you do the coin toss, it says what's the chances that you get a heads 3 times in a row. You'd have to say it's half a chance the first time I toss it, it's half a chance the second time I toss it, and it's half a chance the third time I toss it, because it is a 50-50 to get heads or tails. So when they say in a row, you just multiply your chances and the results of your chances that many times. If they say 3 times, and that tells you that you can multiply this and say it's a 1/8 chance that I would get heads from a coin toss 3 times in a row. So when we will see examples where we do have to multiply if they do ask you about that. Okay, for monohybrid practice today, I'm going to work through these questions with you so that you're prepared to do the following assignments successfully by yourself. And the first one, you just have to look at what they're asking you here. Homozygous dominant, I'll do this one first, times homozygous recessive. And we're looking at tall and short. And whenever they describe the genes that we're using for the cross, I always say do a legend and have that handy. And they tell you here that there's tall that's dominant and there's short that's recessive. Usually I suggest putting those in a box, and you have that to refer back to as you do your cross. So for A, they're saying that homozygous dominant, homo meaning the same, you have a homozygous tall plant and you're crossing it with a homozygous recessive plant. That's the cross we're doing for number 1. And I'll do it below in a moment. For B, it's asking you to cross same genes, but this time heterozygous plant that's got a tall and a short, crossed with another heterozygous plant. And they're asking you what the offspring would be with that cross. The next one is asking you what happens if you have a homozygous dominant crossed with a heterozygous plant. And D is asking what happens if you have a heterozygous plant times a homozygous recessive. So these are all different scenarios. We're going to go below and try them out, and you'll see how easy these are to do. The homozygous dominant here is crossed in A with the homozygous recessive. And remember, my legend was saying that this is the tall plant, this is the short plant, and you can see that this plant only had tall genes in all of its, like, let's say sperm, and this one had all short genes in its eggs. So no matter which sperm and egg got together, the offspring are all heterozygous. So in this case, you would say that the babies are all 100% tall for the physical, for the phenotype, for physically, they're 100% tall. If they did ask you genotype, you'd say they would be, for gene types, 100% big T, little t. And your first question is done. For B, the question was heterozygous for one parent and heterozygous for the other parent. So in trying out the possible offspring for this, you'll see that what you get physically is, phenotypically, is that you get 3/4 that will be tall and 1/4 that will be short. So you could say this is a 3-to-1 ratio of 3 that would be tall to 1/4 of their offspring that would be short. So if they did have, for example, 100 offspring, it would be about 75% of them that would be tall plants and about 25% that would be short plants. Let's go to C, which was a tall plant crossed with a heterozygous plant, homozygous tall with heterozygous plant. And you're just going to check out the gene combinations here. And in this case, you're getting 100% that are tall. Now, these are homozygous tall, these are heterozygous tall, but if they're asking you physically what they would look like, you would have to say they're 100% going to be tall plants. If you were looking at these in your garden, you could not tell the difference between a homozygous genetically tall plant or a heterozygous genetically tall plant. If they asked you about genotype of your offspring, then you could say it would be a 1-to-1 ratio of tall, pure tall, homozygous tall, and heterozygous tall. but we're going to stick to phenotypes, I think, for this question. Okay, and then the last one was what would we get from a heterozygous and a homozygous recessive plant? And you can see that these are easy to do. In this case, you're going to get a 1-to-1 ratio physically of, we don't say 2-to-2, we say 1-to-1, so we reduce it to the lowest possible ratio, a 1-to-1 ratio of half of them being tall, that would be these guys, and half of them being short, that would be these guys. And that is your answers to those questions. On number 2, we'll work through this one before I send you off on your way to try some. This one is talking about cystic fibrosis that is recessive, and so the normal would be the dominant. So I would use, you can use any letter as long as you follow your little legend. I'm going to use N for normal gene and little n for the cystic fibrosis gene, and that will be my legend for this cross. You can use other letters if you like. But this question says that both parents are carriers. So both parents are, normal, but they both are carrying a recessive gene for cystic fibrosis. So when we try this question, I'll just go down below here and give it a try. This is 2A. When we cross these two parents, both who carry a gene for cystic fibrosis, you can see that their chances of having a child with cystic fibrosis is going to be 25%, and having a child with cystic fibrosis. And the next question says, what's the chances of it being a boy with cystic fibrosis? So this is the probability thing that we're going to do. They've got a 1/4 chance of it being cystic fibrosis, and the chances of a boy is always 50-50. So the chances of a boy is 50%. So this is how you multiply the tops, multiply the bottoms, and the answer to the question is that they have a 1/8 chance of having a boy who has cystic fibrosis from this cross, because it's a 25% chance of it having it, but half of those would be boys. If the question was, I believe there was a question about 2 in a row with cystic fibrosis, that's just going back to this original cross and saying they have a 1/4 chance. So in answering C, if they have a 1/4 chance, and the question was, what's the chances of having 2 in a row of cystic fibrosis children of any kind? You would say it's a 1/4 chance the first time they have a baby, and it's a 1/4 chance the second time they have a baby. So their chances are you multiply the tops, multiply the bottoms. It's a 1/16 chance that they would have 2 in a row with cystic fibrosis. And that is your answer for C. All right. We are going to see these are things to omit here because I took those questions out of your notes. Before I send you off, I have one more thing to just mention about if you wanted to check if your organism, plant, sheep, whatever, is actually purely dominant or heterozygous, and if you are choosing something like the male to breed your herd with, you may want to know that it's dominant for a certain trait. The way that they test this is always tested against a homozygous recessive individual. And so you can see here, if they are totally dominant, all offspring will still be dominant, even if they cross with a recessive, because that recessive will be hidden. And so using a homozygous recessive, you can tell quickly after breeding it whether there was any recessive being carried in that individual. Because if there was, if you're trying out crossing your, let's say your animal, and you cross it with homozygous recessive, if it was carrying or hiding that recessive gene, half the offspring would actually be recessive. And so a test cross is where you cross your individuals with a homozygous recessive, and then you're going to be able to look at the offspring and see whether in fact there was a hidden recessive in that individual, because half of the offspring would end up recessive. And this is a real life example with sheep. If you have a sheep that is white and you want white wool because you can dye it, there's a bigger market in selling it, you want to check if your sheep is actually purely white or carrying the recessive. By the way, if your symbol is the same looking symbol as the dominant, my suggestion is put a line over it, and it will keep you from ever making errors in your crosses. So this is a homozygous dominant white sheep. The possibility looking at the sheep is that it may be carrying a recessive gene for black. This little black sheep is the one we're going to cross it with, and that way we can tell what the gene of this one is. So here's our possibility that it's pure dominant white. We cross it with our little black sheep, and we see that if it was purely dominant, all babies would be white. Over here, if we see it has a if it has a hidden black gene and we cross it with our test cross, our homozygous recessive black sheep, half of those babies would be black and half the offspring. And so it's very easy to do these crosses called test crosses. And a test cross means always cross with a homozygous recessive, and then you can quickly see what the genotype of that other sheep was. Okay, so in this case, if all the offspring, if you look at all the offspring and you saw some black ones in there, you knew that the parent would have been this heterozygous parent because some black offspring resulted. If you got only ever white babies, you would know that your parent was pure homozygous dominant if no black babies resulted. Okay, I will send you off to work on page 9 to 12 in your notes, and you can check the key, and happy crossing. We're going to practice the monohybrid crosses, do some